That was fabulous. And thank you very much. Absolutely brilliant summary. Really very informative and fascinating. I mean, I think the one thing I was going to touch on, which to add possibly, is this whole construct of prior therapy and long-term complications from CAR-T. Just my own impression in real-world practice now, and using cilta-cel early, actually, in at least actually second line, actually, is where I've been using it. In other words, after first line fails, then salvage, and then using it subsequent to initial salvage has been, it's so important that less treatment prior to deployment of cilta-cel. I've seen great outcomes in my patients who've been less heavily pretreated, to your point. One remarkable case in a man who was 17p deleted, and after isatuximab KRD relapsed, we then salvaged him with a novel combination of PVD and selinexor, got him back into complete response because he was 17p, and then took him straight to cilta-cel. And during his CRS, day 8, his Tmax was 105. We actually had to put him in a cold bath to stop him from, getting into real trouble with hypothermia. We never transplanted him, and his wife was against it. So she was a children's hospital nurse, and he didn't want to. And he did extremely well. His MRD negative CR looks fantastic. I was just with him about a week ago. Brilliant performance status. It just struck me that I just wondered if we had given him high-dose melphalan prior to the cilta-cel. I'm just not sure how well he would have done with that Tmax of 105, for example. A very interesting case, very informative to me. Anyway, he's done beautifully, and he's 17p deleted. Obviously, it's still early days, about a year out now, but nonetheless, I'm so impressed with how well he's done, and his quality of life has been superb.
Now, we have a quite similar case. We had a patient, which I saw for the first time 4.5 years ago, and he had a really high-risk multiple myeloma. He was treated outside of our center because of high-risk myeloma. He received what is in Europe still standard for high-risk disease, a tandem transplant, but he had a relapse 3 months after the tandem transplant, and he was kind of referred to us with a question whether we would offer this patient an allogeneic stem cell transplantation. And that time we had the first CAR T-cell studies available, and we discussed both options with the patient, and a patient who was 40 years old, so a very young patient. How he actually was very much in favor of CAR T-cell therapy, received the CAR T-cell therapy, and is now in molecular remission since 4 years after the CAR T-cell therapy, which I think really indicates that we have a curative potential with this kind of treatment, even in high-risk disease.
So Herman, this was a brilliant presentation, and there's still a problem, and you pointed that out. That patients with poor risk factors like high ferritin, refractory extramedullary disease, they do poorly with CAR T cells. So what is the next step in those extremely high-risk patients from your perspective?
Yeah. Heinz, this is the $1 million question. What I think is probably these are patients where we then are starting to combine immunotherapies. And I think this is probably a patient where if I had all the treatments available, I would probably give the patient a CAR T-cell product and then kind of consolidate with a bispecific antibody. But I think that is clearly at the moment a hypothesis, and we need the trials to show that this is effective. Yeah, but I think you're completely right. These are the patients where we still have to definitely improve our strategies.
I think Meryl's just raised her hand. Sorry, Meryl, please go ahead. I had a comment to that, if I may, Herman.
And thank you for the opportunity. I think the high-risk patient, the one Herman has just shared with us, tandem transplant followed by CAR T, I think that's a model. That could be a model for such patients, because we know that CAR T cells are not able to overcome high risk, but maybe after a more intensive approach and followed by an immunotherapy, that could be a solution.
The only caution I would put to that, Meral, is this. If you looked at the real-world experience from the Mount Sinai group, it wasn't presented at last year's ASH, but at the ASH the year before, they presented 70 cases of real-world heavily pretreated patients, and they had a 28% incidence of myelodysplasia. And when you looked at those patients, they had all had prior high-dose melphalan. And I'm not saying it, again, I really don't want to beat the drum of the melphalan question inappropriately, but I just want to bring into mind that strategic consideration, because if you can get there without necessarily using the intensity of high-dose alkylation, particularly in the context of tandem effects, I'm just wondering if that's a wiser approach. The second, we don't know, but obviously might be. My second comment, Hyman, was to the ferritin level. Having just been treating patients who've had resistant COVID with CAR-T and myeloma, to the extent it's been life-threatening, I'm struck that we have to be a little bit careful about the sequence of CAR-T and bispecific, especially if you've got endotheliitis and a persistent ferritin level that's high. Because, for example, in the COVID setting, we've learned that that's actually quite risky, especially if they get infected with COVID. And that sort of persistent endothelial signal, I'm just wondering of the wisdom of immediately going to a bispecific. But anyway, just a comment there.
No, I'm completely with you, and I guess we definitely have to wait until we switch to a new to start any immunotherapy which is T-cell based in patients that have high inflammatory markers. I think completely agree with you. I think the likelihood that these patients are going to develop complications is extremely high.
Yeah, I'd like to, because my hand is yellow and my background is yellow, so my hand. Fantastic, Herman. And in Brazil, the cilta-cel was approved 2 years ago, but there is no slot, of course, and probably we will start now in the second semester with cilta-cel in commercial. But let me ask you, because I have seen some doctors to offer maintenance for patients after CAR-T, and do you have an idea which patient we can use maintenance or not? Because I'm very surprised that there are many doctors using maintenance after CAR-T.
No, I think that's a very important question, Vania, and I think for me the philosophy of CAR-T cells was always it's a one-shot treatment, and that's how we actually tell the patients that we tell them it's an intensive treatment, but then after this treatment you are treatment free for several years without any additional therapy. And I think this was one of the reasons why these patients had such a great quality of life after CAR-T cells. And if we actually introduce additional treatment, this will have an impact on their quality of life, a negative impact. On the other hand, we now see quite a lot of patients that are unfortunately also failing CAR-T cell therapy. We know that CAR-T cells persist for very limited time. So, for example, cilta-cel is detectable between 80 and 160 days post CAR-T cell infusion, and then it's gone. And ide-cel probably a little bit longer, but then it's also gone. And there are some data that show that persistence of CAR-T cell is important for long-term disease control. And that's why biologically it probably makes some sense to give some maintenance, but I'm completely with you. I think for the philosophy and for this persisting CAR-T cells, that this one-shot treatment, it's not a good strategy to add additional treatment.
Can I?
Well, yes.
Sorry, Marie. Go ahead.
No. Go ahead.
No, okay. Actually, another situation is about this GPCR5D, I don't have experience, I don't have study with the CAR-T, but we have many studies with the bispecific, and you comment about the cerebellar toxicity, and I have seen cerebellar toxicity. I'd like to know about your experience.
No, unfortunately, we don't have experience with GPRC5D CAR T cells. In the study that was published in New England Journal, there were some cerebral toxicities, and there were some arguments around this, that there was some expression of GPRC5D in areas of the brain that are important for and thus but we have a lot of experience with talquetamab. I think that's what you also have. And we have seen a lot of mucus toxicity. We have seen skin and nail toxicity. We've also seen patients that have completely lost their taste. But I haven't seen with talquetamab the cerebral toxicity. But it was described, and you confirmed that there is some issue around this. So thanks for sharing this with us.
No, I have some patients, and it's difficult because we stopped the talquetamab, and the patient keeps on the. I'd like to know the experience from my colleagues.
Yeah, Vania, I've had the same experience with talquetamab. Some great responses, so I've been very impressed with it. But I've also been struck that I've had some patients who run into real trouble with both the cerebellar toxicity. The other thing is with the mucosal toxicity that Herman mentioned. I see real impressive anorexia and weight loss. I had one lady lose about 20 pounds in 2 months, and she really couldn't afford to. So we actually had to stop therapy because she became, literally, she became cachectic almost. It was very challenging. I guess the only thing I would say about how we've approached it is to try and spread out the infusions, learning from the Jeffrey Mateos paper at ASH, where he combined it with POM and was able to really use pomalidomide as an adjuvant with the talc, and then reduce the frequency of talquetamab use, and therefore reduce the toxicity. That's what we've been trying to embrace. The only other issue we run into is exhaustion. Immune cell exhaustion. In other words, they're just running out of gas from tech. Then you try to move across to talc. That's a strategy we've actually found not very helpful. In other words, even if you switch targets, if they're immune exhausted, you're stuck. So there've been lots of things we are learning, but it's been very exciting, Vania. But again, my sort of philosophy has been we need all hands to the pumps, not just the immune therapies. We need to be able to sequence and drop therapies into the middle of these sequences to restore immune function and to give the immune system a proverbial break as we use other strategies. And obviously cell mods, for example, have been one of our strategies in that space.
If I can just, add a comment. Thank you, Herman, for the presentation. And my question was going to be based on maintenance, that it was raised by Vania. But, my point is a bit different because now in Spain, we are going to have available ID cell in the triple class exposed myeloma. And we know that ID cell finally results in a median PFS of approximately 9-10 months. If the patient is in CR, the durability of the response is going to be longer. But we are going to deal with heavily pretreated myeloma patients. At the same time, we have experience coming from clinical trials in some patients receiving ID cell followed by iberdomide maintenance. And I've seen how in the majority of the patients we put in this clinical study, in spite of the median PFS expected of 8-9 months, iberdomide has been able to prolong the durability of the response. So my idea and my question is if you, in your clinical practice, when you use ID cell in heavily pretreated myeloma patients, in which, of course, the ideal of a single infusion is important, but the addition of lenalidomide, maybe 10-15 milligrams with good tolerability, can help us to maintain the patients for a prolonged period of time on this line of therapy. And this is my question to you, Herman. I don't know if you do this in the clinical practice.
So I think this is a very important question, and Vania has already addressed this, and you are completely right. In heavily pretreated patients, we see limited efficacy, limited duration of response with ID cell monotherapy, and especially in patients that have a suboptimal response or patients that have that are slowly progressing with their multiple myeloma, and we still see CAR T cells in the peripheral blood, then we, in some patients out of any protocol, we have tried either lenalidomide or in some patients also pomalidomide to reduce the hematotoxicity. And we've seen expansion of CAR T cells. So that is an interesting approach. There are also data that show that with proteasome inhibitors, you can also upregulate the expression of BCMA. So that might be also a strategy to improve CAR T cell function. But I think we have to learn much more about this. And I don't think we have already good data that we can say this is the strategy that we should apply.
Okay, and just a very brief question. We discussed just before about GPRC5D bispecific monoclonal antibody. I know that you had some experience with the use of talquetamab as bridging therapy before BCMA CAR T. Can you comment? Because this means that patients will receive just, well, some doses. So what is the efficacy and what is the safety profile? And if you consider that this will be an excellent opportunity in order to go to the CAR T with a low tumor burden.
No, thank you very much for this question. I think what we learned from KarMMa-3 is that an effective bridging therapy is important for a good success of a CAR T cell therapy. So in a patient who has an increasing tumor load during the waiting time until the patient receives the CAR T cell, this patient is doing extremely poorly. On the other hand, we don't want to have a cytopenic patient at the time of CAR T cell infusion. And you are completely right. We have some experience with talquetamab. We use talquetamab after the leukapheresis so that it doesn't interfere with the T cells that we are obtaining for CAR T cell production. But following the leukapheresis, we start with talquetamab. We see the usual side effects, but what we don't see is that these patients go into severe cytopenia, and we have seen nice responses, and we didn't see any issues with the consecutive infusion of the CAR T cell product.
Yeah, Mary, we have one real world case just to share with you, which is really, I think, sobering. We love that strategy, Herman, too. We've been using TALC to on-ramp for cilta-cel. One of my partner's patients just recently died of HLH after cilta-cel, right in the context after TALC on-ramping. So I think we just have to be a little bit careful when you absolutely rev up the immune system, because he died within days once the HLH declared itself. Declared itself. So just a note of caution there. I completely agree, Herman. It's a wonderful strategy, but that caution. And my second comment was to your maintenance question. My partner Omar Nadim is leading a trial at our center of ELO-IBA post-ida-cel. So ELO plus IBA in the context of natural killer cell leveraging. So what we have done in our group off protocol for standard of care is we've used ELO-POM as a strategy after CAR T for higher risk patients, just on the rationale that NK cells may matter as well.
Sorry, but about the GPRC5D, the talquetamab during the bridging therapy, for how long? During the process, or?
It's only between the leukapheresis and the CAR T cell infusion, so it's only for a few weeks. Meral has a question.
Yeah, so I think Vania and me, we both share the issue of access to CAR T. And since there are two groups here, the Spanish and the German, who produce academic CAR T. And so my question is, how do you compare the commercial versus the academic in terms of future accessibility? And we have seen the efficacy data from the Barcelona group, and waiting eagerly for the Caramba. And so I would like to hear your opinion in any kind of future for us.
Ladies first.
Okay, so from my perspective, I think that for us is a great opportunity. This is what I have to say, because we are not so rapid as Germany in terms of reimbursement for Einsele and Filtacell. And the academic CAR-T is right now our great opportunity to deliver BCMA CAR-T in relapsed refractory myeloma patients after 2 prior lines of therapy if they are triple class exposed. So this is the main advantage. But the second one is the flexibility, because we are in continuous contact with the Clinic of Barcelona. So if we have an urgent patient, they will do the space in order to allocate the manufacturing process. If the patient is coming sometimes with a rapid relapse and we ask, they are able to release the product. So the flexibility for me is relevant. And another important point, the academic CAR-T allow us to freeze the T lymphocytes. And we have some patients in which, while the relapse is very aggressive, we decided to start by GPRC5D because of the aggressiveness of the disease, but we have collected previously the T lymphocytes. So the T lymphocytes are frozen in our center, and just in case, if it is necessary, we will reutilize later on. And in terms of efficacy, I would say that it is not so effective as Filtacell, but it is a bit more effective than Einsele. And well, our experience is good, but the most advantage from my point of view is the flexibility and the availability, that it is definitely easier than with the commercial ones.
Yeah, I must say Spain is far, far ahead of Germany in their academic CAR-T production. So we have about 3 or 4 products that are explored in still phase 1-2 trials. And we would never, none of these centers that are using these academic CARs would be able to cover the whole country with academic CARs. And we haven't reached with all these trials any efficacy when compared to the Spanish one. So if you want to learn about academically produced CAR T cells, Spain is the first address in Europe, I guess.